Hello, my name is Ryan Salazar, and today we're going to be talking about genetics.
Kane is in the building.
This figure shows us an example of DNA synthesis in eukaryotes. In figure A, we can see that the initiator protein has already opened the double-stranded DNA molecule. DNA helicase then begins to unwind the molecule further. In figure B, the RNA primer initiates synthesis of a new DNA molecule called the leading strand. DNA polymerase III then continues to elongate the new DNA strand. Figure C shows the second single-stranded DNA molecule being made and is called the lagging strand. Here, there are many RNA primers involved, and when each primer is removed, it leaves a little nick in the DNA called an Okazaki fragment. These have to be fixed by a protein called DNA ligase. After both the leading and lagging strands have reached the end of the template DNA, the process is complete and two double-stranded DNA molecules are left. Now we are going to talk about eukaryotic transcription. Transcription is similar to DNA synthesis in the fact that a new molecule is formed from a DNA strand. Instead of making a new DNA molecule, however, we make a molecule of RNA. To do so, we start with a protein called RNA polymerase. RNA polymerase binds to a region of DNA called a promoter and begins RNA synthesis. The protein moves down the DNA molecule. Notice that there is only one strand of DNA being copied. This is called a template strand. Another difference to note is that there is no DNA helicase this time that keeps unwinding the DNA. The DNA reforms its double helix as the RNA polymerase moves down the molecule. After the desired DNA code is copied, the RNA transcript being created is released, and the RNA polymerase detaches from the DNA. Note that this entire process happens in the nucleus of a eukaryotic cell, and that only a single-stranded molecule of RNA is made. The last topic we're going to talk about today for eukaryotes is translation. Translation is the process of making a protein from reading mRNA. Remember the mRNA we made from transcription? In translation, we use a molecule called a ribosome to read the mRNA and make the desired protein. This happens in 3 steps. The first step is called initiation. Here, the ribosome, known as the 30S initiation complex, surrounds the mRNA along with the transfer RNA, or tRNA. Notice that the tRNA carries the amino acid methionine. This is because the codon for methionine is the start codon in translation. Elongation is the second step in translation. This is when the protein is being made. The ribosome moves down the mRNA one codon at a time, adding another amino acid to the growing chain each time it moves down the mRNA. A protein called peptidyltransferase binds the amino acids together. It forms a peptide bond between each amino acid. This happens as the tRNA connects to the mRNA and passes through the ribosome. Remember that tRNA is the RNA that is holding the amino acid. Termination is the final step in translation. The ribosome hits a nonsense codon in the mRNA, and a release factor binds to the codon. The ribosome separates from the mRNA. The mRNA and the newly formed protein are freed, and the process is complete.